Hi all. Hi all. Remember this important points regarding the topic cell cycle. So mostly in meiosis, we will be getting question under prophase 1. So we have to first of all remember there are 5 phases in prophase 1. Nothing but leptotin, zygotin, pachytin, diplotin and diakinases. So here we should remember what are all the process happen exactly at leptotin. So leptotin is the very first stage where the chromosomes condensation starts, and the chromosomes will seems to be a long, slender chromatins. The chromosomes condensation starts at this phase, whereas zygote, where the chromosome condensate, the condensation will become more. We can say more condensed form of chromosomes will be found. Then the similar chromatids of each chromosome. When we make chromosomes, we put 4 arms. Each arm is copy of one another. So we can call this as sister chromatids. Since we have a pair of chromosome, this is nothing but homologous chromosome. Here each chromosome is a bivalent. We can say now the bivalents were formed. That's what I am meaning here. The similar chromatids will come for pairing. The similar chromatids will come for pairing. To make them pair, there is a protein involved here. The protein is called as synaptonemal complex. Synaptonemal complex. So if synaptonemal complex comes, then the bivalents of each chromosomes can come together and form a structure called synapses. So that I can show you here. Since I made 2 different color chromosomes, this is one homologous pair. So these chromosomes will join at some point. Either we can mention it like this, or to make it some more clear, we can say the chromosomes will totally joins at some point. Who makes them join means the synaptonemal complex makes them to join, and wherever the point where they joined, they are called as synapses. Maybe I can just leave it here. Now I can show you there is a point where synapses were occurred. Now after making the synapses, if we go to the pachytin stage, this pachytin stage is very important. The very first statement we have to remember, this stage is comparatively longer. The stage is comparatively longer, where recombination nodules occurs at this stage. Recombination nodules occurs at this stage, and the chromosomes will do crossing over. 3 terms you have to remember now. Number 1, either we can call it as crossing over or exchange of chromosomal material, chromosomal materials, or we can also call it as recombination. And this is the reason for variation out of sexual reproduction. So at pachytin, which is comparatively a longer phase, where recombination nodules occurs, because of that, either we can say crossing over, or we can say exchange of chromosomal material, or recombination occurs. Where it occurs in a homologous pair between the non-cistral chromatids. This is very important. Between the non-cistral chromatids with the help of an enzyme. All metabolic reactions require enzyme, right? The enzyme involved here is recombinase. Remember, word ends with AAC is the enzyme. So, some part of chromosomes from one chromosome, some part will come to this side, some part will go to the other side. So, this is what we call exchange of chromosomal material or crossing over, or also called recombination. Now, at the end of this, the cell will go to the next stage called diploting. At diploting, first of all, you should remember the synaptonemal complex which is formed at leptotin, right? So here it will undergo dissolution. That means nothing but disappearance. So dissolution of synaptonemal complex occurs. Then the homologous chromosomes will come to a stage. The homologous chromosomes will come to a stage called chiasmata. Chiasmata. This represents the alphabet X. So we can say the X-shaped structure of chromosome after crossing over, which happened at Paquetin, right? At that point alone, chromosome will remain together. All other junctions, for example, in my picture, I have mentioned there is two synaptonemal complex. The upper segment has not done any crossing over. So that part will come out. That part will open. Whereas the part where exchange has happened, that part alone will remain. The lower part, this part alone will remain as it is. This is what we say X-shaped structure. If I complete this picture, in fact, it will come out like this. So we have to make one of the homologous chromosome like this, and other homologous chromosome, I have to cut it here and make the other chromosome properly. Let me take the opposite color. So let me complete this chromosome with blue color, and let me draw the another member of this homologous chromosome, and see now how I'm going to complete. I'm completing it with red color. So earlier, because of synaptonemal complex, synapsis has happened. Now, because of enzyme called recombinase, which I mentioned here, the exchange, crossing over, recombination happened. So what happened now? Observe this. So a part of red chromosome came to other side. I can put one line here, and a part of blue chromosome went to other side. So I can mention it a blue color line. So now how it looks like? It looks like an alphabet Z. The upper part where the crossing over has not occurred, that part will open. That is what I mean here, dissolution of synaptonemal complex. The part where exchange occurred, that we are calling as a chiasmata. More importantly, at this stage, in oocytes of some vertebrates, not all vertebrates, remember it might come in a statement based question. In some vertebrates, this stage might last for months to year. So at this stage, cell might get arrested for months to year only in oocytes of vertebrates. So oocytes nothing but oogenesis process. Now, when the next stage, diakinesis comes. So now we should remember the chiasmata has to open. So now I have to mention that the chiasmata will end up. That's what the term says, terminalization of. Terminalization of chiasmata. That means even the junction where the chromosome attach, they also will go out. Now see this picture. The junction where the chromosome attach, that also will go out. So now I can say both the homologous chromosomes got totally separated because of the chiasmata has been terminalized. That's what we are meaning here. The term diakinesis, di means two, right? Kinesis means breaking, right? The homologous chromosomes are separating now. Now the chromosome condensation will complete. Chromosome condensation completes. Remember, we told condensation started at left protein stage. Condensation started, but it will not complete. Once if condensation completes by forming a complete chromosome, then where the recombination will occur? At the junction of recombination, the chromosomes, nothing but DNA only, right? It should be loose enough to form synapses to do recombination. Except that part where recombination occur, other segments can tighten, nothing but contains. That is why synaptic complex go out at the stage called diplotin. Then when diakinesis come, that chiasmata is also going out. So the point where exchange occurred, that will condense totally now. So we can say chromosome condensation completes. Then the spindle fibers. Spindle fibers will come, which is originated from the centromere, that is the two poles of the cell. Further, the nucleolus and nuclear envelope will disappear. Nucleolus and nuclear envelope, nothing but membrane. Nuclear membrane will disappear. So, learning about this 5 point is very important under Prophase 1.